Because Erickson, are you accepting carbon?
I crossed that out and circled what you wrote previously.
Oh, perfect. Because I could not remember if it was carbon or CO2.
Okay. Part 3 of cellular respiration is the ETC, also known as the electron transport chain, and then chemiosmosis. And so, again, tomorrow you'll have a quiz on the ETC. It's the figure in your book. I think it's 7.12. I don't know, something around there. But it is posted on Google Classroom what figure it is to study for. Oh, God, I feel like there's going to be some sound. Better mute this.
Some sound.
Okay. No, what? All my slides went blank again. Are you kidding me? Why does this always happen?
Suspicious packages.
I hope you get sick.
There we go. Okay, so in our journey of cellular respiration, we started with glycolysis, taking glucose, splitting it into 2 3-carbon molecules called pyruvate, generating some electron carriers that will end up in ETC. So we're actually going to figure out what happens to them. 2 total net ATP produced, and then we have that shuttling over with the acetyl-CoA, entering the Krebs cycle again to ATP. Bunch of electron carriers. That's the purpose of the Krebs cycle. And so today we're going to focus on the ETC and how we generate ATP through oxidative phosphorylation. Both of these have been substrate, and now oxidative is a little bit different, and we can generate a whole bunch of ATP. So looking at the ATP accounting so far, glycolysis 2, Krebs cycle 2, only 4 ATP. There's got to be a better way. A little fun fact here: the working muscle recycles over 10 million ATPs per second.
And those only make 2.
Yeah. So there's got to be a better way, and there is. It is the ETC. And so this happens in the mitochondrial membrane along the cristae, and there's just a bunch of transport proteins and enzymes that work together to shuttle across hydrogen protons, or we'll just say protons, build up a proton gradient, and then it's going to go through an enzyme called the Atp Synthase to make approximately 34 to 38 ATP. The number is not exact, and this is just from one molecule of glucose. The only way that this happens is that there's got to be oxygen for it to occur. So this is aerobic respiration. The other processes, like glycolysis, oxygen is not needed. So glycolysis happens no matter what.
So it could be aerobic or anaerobic? Doesn't matter.
For glycolysis?
Yeah.
Yeah, because it just happens all the time, but you don't get a lot of ATP.
And same with Krebs cycle, it's both.
Yes. Yep. Okay, so with the mitochondria, looking at the structure of it, it is a double membrane. Remember from endosymbiosis theory, how an early archaea of some sort got swallowed by a eukarya and they live in symbiosis together. But we have an outer membrane and an inner membrane, and so these highly folded areas are the cristae, and this is where we're going to find the enzymes and transport proteins lined up. And we also have an intermembrane space, which is just the fluid space between membranes. So looking at, up close, the double membrane inside the mitochondria, we have the intermembrane space, but it's still in the mitochondria, and then we have the mitochondrial matrix. And so here are these proteins that are bedded in to this membrane, and what's going to happen is electron carriers are going to drop off their electrons, and these electrons are going to get carried by other proteins and enzymes and travel through. And as they travel through these proteins, hydrogen ions will be pumped across to build up a gradient. So remember those electron carriers from both glycolysis and the Krebs cycle, where we can generate 4 NADHs, 8 over here, 2 FADHs. Well, it's time to break open the piggy banks and release those electrons so they can do some work for us. So this is where we'll start here. So our electron carriers, NADH and FADH, have these electrons, and they're going to drop them off at the ETC. The first enzyme that picks up the electrons is NADH dehydrogenase, and the carrier that picks up these electrons is called ubiquitin. And I think your book abbreviates it as, I think it's Q, but don't quote me on that, but it's ubiquitin. So what happens is that these electrons get shuttled to another set of enzymes called the BC1 complex, and then it's going to get shuttled to some more enzymes and proteins. But this is the first kind of proton pump that that drives protons to the inner membrane space. Then the third set is the cytochrome oxidase complex, where it gets picked up by cytochrome C, and I think they just abbreviate it as C, so it's Q and C. And NADH gives electrons to the NADH dehydrogenase. This is where
See, we can't see the whole board.
sorry.
And we also have another question. What does this cycle look like in our book?
All right, it is figure 7.13. This is what it looks like. Oh, hold on, let me fix the board still. I haven't done that. Good gravy. Zoom.
So this one's more of a line than like a rotation.
Okay. So it's flipped. So this is the mitochondrial matrix. This is the inner membrane space. So it's flipped from the previous diagram. So here's the electron carrier. Drops off the electrons to the NADH dehydrogenase. Gets picked up by ubiquitin, abbreviated Q. Shuttles it to the BC1 complex. As the electrons pass through these complexes, protons get pumped across. So then it gets dropped off at the BC1 complex, where it gets picked up by cytochrome C. Then with cytochrome C, it goes to the cytochrome oxidase complex. More protons get pumped across. We'll talk about this, but water is formed. So this is a byproduct of cellular respiration, H2O, because it's a combustion reaction. But those protons, as well as oxygen, this is why oxygen is needed, is the final electron carrier. So that's why oxygen is needed for this to happen. So 2 protons and an oxygen create your water. And then what happens with these protons is somewhere further down the line, we have this huge complex called the ATP synthase. And so these protons go on through, kind of like water through a turbine or wind going through a turbine, but this thing spins and literally takes ADP plus P and puts it together to get ATP. And then the whole process kind of repeats itself, where these hydrogens will float back on over here and get pumped across again.
So is the part on the right part of the diagram as well?
Yes. So this is the ETC and chemiosmosis. And that's why it's like osmosis, because it's not water, but water is involved in it. So, and it's a chemical. So I don't know. It's diffusion. It's going from high to low to generate this. Okay. Not now. Okay. So this is what it looks like. Back to this cartoon diagram. NADH drops off the electrons, gets reduced. These get shuttled back to the Krebs cycle and glycolysis. The electrons travel through the NADH dehydrogenase, pumping hydrogens across, gets picked up by ubiquitin. This is where the second electron carrier drops off its electrons. It also gets picked up by ubiquitin, so it bypasses this first protein. Ubiquitin travels to the cytochrome BC1 complex. More hydrogens get pumped across. Electrons get picked up by cytochrome C, which travels to the cytochrome C oxidase complex. Hydrogens get pumped across. And then these electrons, after they've traveled through these 3 complexes, get picked up by oxygen. That's the final electron carrier, combined with 2 H pluses to form water. Woo, yeah.
2 things. So first off, is this kind of like the opposite of the other 2 then, in the fact that the electron transport are like actually like doing the dropping off instead of getting made?
Yep.
And then what, half O2? Wouldn't that just be 1 O?
Well, because it goes through the cycle twice. Remember, we still have 2 molecules of pyruvate, so then we have like 2 of everything.
Oh, so really it is O2.
It's O2, yeah, but
it's like after twice, it's
Yeah.
O2.
All right, so basically you're just dropping off electrons, building up that proton gradient, and it's going to go through that ATP synthase. So electrons are stripped from these H atoms. That's why it's just H plus, and they travel through the ETC, building up, again, that proton gradient. So electrons are doing a lot of work here with their energy. And then this is what pulls the hydrogens across. It's just diffusion, okay? You're moving from high to low, and you're building up this, oops, I just said that wrong, didn't I? Yeah, okay. We're building up, so low to high here. So that's why you need the electrons for energy. But the electrons are really traveling downhill. And that's because this is a high energy carrier, and then it just like gradually gets lower energy-wise. So electrons flow downhill. In reality, if you were to look at energy level, according to this free energy, and then I don't know what this is, but it's got the proteins, there's actually a lot of other enzymes involved that you don't need to memorize, just the big ones. And so these electrons are literally just being shuttled through. So like this says FMN to iron sulfur to ubiquitin to cytochrome B to iron sulfur to cytochrome C1 to cytochrome C to cytochrome A to cytochrome A2. So there's a lot of other players involved, but you just need to know the 3 big ones and then ubiquitin and cytochrome C. Yeah. So it's more complex than what you actually have to know. But this gradient forms as electrons move through these electron carriers and energy is being released, so that way those proteins can do work and pump hydrogens across. And then finally you get to the chemiosmosis part where you get to produce ATP. And so when hydrogen's being pumped across, you do lose a little bit of hydrogens back through due to diffusion, because that just happens going from high to low, but it's pretty impermeable. I mean, it's not as leaky as it is. So in order for those protons to re-enter the matrix, it has to be through the ATP synthase, and that's the turbine, through the process of chemiosmosis. Okay, so here's what ATP synthase looks like. On your exam, you do need to know what ATP is made up, ATP synthase is made up of. So we've set up a hydrogen gradient. It flows through, it spins. ADP plus P equals ATP. This is chemiosmosis, diffusion of ions across a membrane of some type. Okay, this is the slide that I think is most important. You need to know that ATP synthase is made up of an F0 membrane-bound complex and an F1 complex. Okay, so I don't know how you want to star it, or you want to quickly write it down. F0, F1 complex make up ATP synthase. So I think I have a diagram on the next slide that shows you.
Is that literally like 4 iron? Like the F? Is that something more iron?
Nope, it does not.
Okay.
No. Actually, I don't know what the F stands for. So this is the F0 part, and then this is the F1, the knob and the stop, basically. So very similar to a watermill. Yeah. Now, inside the mitochondria, you have tiny little folds. So I know in all of our diagrams, they show it as like a straight line. Well, that's not the case. It's more wavy and folded. It's not as nice as the diagram shows. So I just like this diagram because it shows you that, hey, here's a kink or fold inside the mitochondria, and you have all these proteins, and it's, they're all just working together to generate a lot of ATP. And as you can see here, it says 32 ATP. It's around 30 ATP. So I think on your exam, there's a multiple choice question that says, how many ATP are made in the process of, cellular respiration or whatever. And you just have to, it's going to be the one in the 30s. I have like 20, 22, 28, and then there's one that's like 36. So.
Yeah, because isn't it like 32 is from this, and then
you add the others, yeah.
And then the glycolysis makes 2.
Mm-hmm. So looking at the theoretical yield of eukaryotes, it's around 36 ATP per glucose molecule. And this is kind of the math portion of it. They break it down due to electron carrier accounting. So you generate 10 overall, 2 from glycolysis, 2 from the oxidation of pyruvate, 6 in the Krebs cycle, and then you also get the 2 FADH2 from the Krebs. Looking at the ATP account in 2 from glycolysis, 2 from Krebs, and then they count the NADH. So 10 times 3 is 30 ATP from just NADH. 2 times 2 is from 4 for the FADH, and then that's just total for these two. So grand total approximately 38. I don't know, 36 to 38. So.
Was there some?
Yep.
So the Krebs cycle happens twice.
Yep.
The ETC happens twice.
Well, it happens with every single electron carrier.
Okay.
Because you notice that it just said NADH and then FADH. It didn't say 10 NADHs. So you have to think about that for every single electron carrier that goes through.
Yeah.
So if I go back a slide,
So it happens twice for everyone that goes through.
yeah. So you're looking at 10 times here, 10, 12
So 12 times to every 2 for
For
the
the
Krebs.
electron transport?
just for the ETCs, yeah.
Yeah, well, I mean, like the ETC happens 12 times, to the Krebs is 2,
Mm-hmm.
to the glycolysis is 1. This glycolysis just happened once.
Once, yeah.
So 1, 2, 12.
Okay, so the actual yield for eukaryotes is 30, and the reason for that is, well, in glycolysis, it needs to be transported to the mitochondria by active transport. So you have to burn a little bit of energy, so you reduce the theoretical yield in eukaryotes. It's lower than 36 for other reasons. Like I said, the mitochondrial membrane's leaky, so you lose some protons. They kind of diffuse back through. And so that's why this is actually a more accurate picture. 2.5 ATP per NADH molecule to 1.5 per FADH rather than 2. So the new number is around 30, but again, just pick the one that's in the 30s. So summary of cellular respiration. Where does the glucose come from?
Well, just
No, glucose starts
glucose
glycolysis.
starts glycolysis.
It's
That's
there.
It's just there. You mean to tell me your cells just have glucose?
sugar.
Well, sugar, and how do you get sugar?
Oh,
you
eating.
eat it, okay? All right, where does the oxygen
You
come
breathe
from?
in.
You breathe it in. Where did the CO2 come from? So what process creates CO2?
Krebs.
The Krebs, okay? Where does that CO2 go?
Out and about.
Out and about, you exhale it,
Yeah.
okay?
I was thinking
Where does the water come from? What process?
the last ETC.
The ETC. And then where does ATP come from?
All
All of them.
So what was the first one? That do
So
they all happen at the same time, or does it go like down a line?
Well,
It's glycolysis.
okay.
And then Krebs, and then
we teach it like it's linear, but it's like happening all the time. It's
because I was gonna say
continuous.
how if the first water didn't come until the ETC, then how did the old glycolysis happen?
All right.
We cracked the code.
So you got the Krebs, the ETC chemiosmosis quiz tomorrow, figure 7.13, and then we'll wrap up Chapter 7 exam on Friday. Do we want to do? Is Herman Norcross? Are you guys still there? Or are you leaving right now?
Oh, I see some kid.
I think someone just walked out of the room.
They just left.
They, as
No.
they-
Never mind. I'll mention it tomorrow.
They listened to you ask the question and then walked.